Hi, my name is Marcos Edgar Herkenhoff. I'm researching on the field of molecular biology and beer. And my lecture today is entitled Exploring the Molecular Landscape of Catharina Sour Beer Fusion of Omics Strategy for Starting Probiotic Strains Assessment. This research was financed by FAPESP and it's attached by FORK. So, this is actually a work that was published recently by the Food Research International, entitled "The Combination of Omics Strategy to Evaluate Starting Probiotic Strain Catarina Sour Beer Style, Brazilian Style Beer." So, if you want to go further in this or a deep knowledge about this research, you can find in this article. So this article was my research is performing in USP, University of São Paulo, and by FORK. FORK is Food Research Center, and this is a center that is a research center from food sciences and food technologies attached by University of São Paulo, and also by applied Technical University of Applied Science in Germany. So to contextualize you, the consumption of beers increased worldwide. This is also helps awaken interest of different beers with different flavors and styles, which includes and highlight sour beers. So the brewery boom is it's helping to another traditions to be growing, like Australian beer, Italy beer, Japan beer, and Brazil. So in Australia, we have the Australian Pale Ale, and that's a country that they are harvest a lot of good hops with different flavors and aroma. In Italy, they invented a kind of mixture with beer and wine, and this is the both fermentations. In Japan, we also have now a kind of different hope that smells and tastes like lime. It's pretty different. And in Brazil, we have developed a style known as Catharina Sour, and that is why we call these four different countries as a new emerging tradition of beer. Catarina Sour is a beer that is fermented by lactobacillus and usually use fruit or another kind of herb to ferment it together with the probiotic and the yeast. And now this style of beer was rising in 2016 and is now a lot of trademarks in Brazil. It's become very popular. We can find this kind of beer with different fruits, for example, passion fruit, peach, tangerine, and grape also, cupuaçu, which is a natural fruit from Brazil. So the main question of this work is if lactobacillus is used to brew Catarina sour, why not use probiotic strains? So to get a beer more healthy or a beer with functional properties. So probiotics in 2001, the World Health Organization defined probiotics as living organisms that when administered in adequate doses confer benefits to the health of the host. A definition officially accepted. So until this, since uh, these days, we are now use this definition for probiotics, and almost this probiotics is from a group or a genera or all genera known as lactobacillus. So the main objective of this study is to develop and evaluate the potential of the Catarina sour beer style as a probiotic beverage. For this, we use microbiology growth count by plate and PM2 PCR. Special profile of genes that confer resistance to hops, for example, or A or B or C, hit A, BSR-A, and REC-A, protein compositions, metabolomics, and flavorings. So it was developed almost, it was developed 15 different formulations with these parameters, not so much hope because the bacteria have problems to survive in a lot of amount of hope. And using these three kind of fruits, peach, passion fruit, and control. Control means that it's nothing. Actually, Catharina sour must to be brewing with a fruit, but we use a encounter without fruit to see the expression of the genes and how these microorganisms can survive or not in this situation with or without fruit. Is if this fruit has some kind of influence. So when you use Streptococcus thermophilus, Lacticase Bacillus paracase F19, Lactobacillus paracase 431, that's also become from Christian Hansen from Denmark. And so how I said before, we developed 15 formulations. We use 9 of these formulations with a single microorganism, and 6 of them you can see in the right side with the starter TH4 together with another probiotic, for example, F90 or 431. And there was preparation, so we use actually a natural process from Catharina sour to brew. So after the cooling, we perform first you can see in the middle of this figure, LAB inoculations. That's which means that we perform the first fermentation of an acid lactic fermentation, and that go forward with a yeast inoculation and another steps from a beer production and maturation, and to the end is carbonation in store, and we also evaluate the survival of this microorganism during the storage time. So for material and methods, then we perform a microbiology count by plate and PM-qPCR by ABI real-time system. Special profiles of genes using also this ABI real-time system. The proteome analysis was performed by the liquid chromatography tandem mass spectrometry, LSMS, and the metabolomic and flavor components analysis by headspace, solid phase, microextraction, HSPME, coupled with gas chromatographic mass spectrometry, GCMS. So our first results show that you can see this is all the formulations counts by the day 28, which is the end of the fermentation, and day 58, which is the end of storage. And you can compare here in the columns on the middle by per plate, far in the right by PM-qPCR. So generally you can say that PM-qPCR shall grow much more big count. So it can be because normally the cells from the bacteria are stressed, and so they let to demand much more energy and also time to grow good, even in a good culture medium. So that is why PMA qPCR show the best results. So here in this gene expression, so how we can imagine control in passion fruit show a different partner of expression because these formulations has had RC gene as more expressed in the other way. Peach fruit showed that hit A is the most expressed gene. This data is pretty interesting because it's not necessary that is not a key gene involved in that, but sometimes can be RC or sometimes can be hit A, for example. It is what this data show. So by proteome, uh, that is the web from our protein interaction, but it's from the yeast. And by the formulation of TH4 and F19, you can compare that it's not so much difference in the tree, including 431, control passion fruit or peach, day 2, because it's only compared day 2 because the yeast was inoculated by the wharf after the first fermentation, the acid lactic fermentation, and it was performed for 24 hours. So that's why it's only the second day, and you can compare it, and it's no difference by the yeast, but yes, some a little bit more, a little bit less, but it's not so much, which means that it's always okay with the yeast fermentations not interfering. The bacteria are not interfering in this fermentation, which is important because it's necessary for produce beer. So, by comparing the proteome analysis of the microorganism is interesting because TH4 is not performing any networking with the proteins, just proteins related to transcriptional factors, which is interesting because it was comparing the day 1 and with the day 2, but almost in the samples from TH4, the pH, which is indicator for fermentation, was not lowering as so fast like F90 in 431, which can explain this absence from glucose or another sugar metabolism in the first day of TH4. TH4 seems that is not, it's a little bit delayed compared to F9. F90 and 431, you can see that in this network, and it's more related to sugar metabolism and survival of this microorganism, and so for the fermenting the worth what we expected. So for 131 also the same situation, just a little bit different, not so okay. That's for example for control day 2 is a little bit small, but there is activity just is so much different than TH4. The results from the flavor omics analysis show that it's difference in the the production and the synthesis of some kind of compounds, especially these volatile compounds. And so TH4 or F90 or 431, that's showing here that it's some compounds that's the same, okay, but this data show that it's all different. So the strain that you can that it's used from beer from brewery can influence also the strong the sensorial aspects, and so it's not so the bacteria is interesting because it's not so complex microorganisms. It happened almost with the yeast. Some kind of yeast strains has so special flavor and so special odor, so confer a really different uh, sensorial uh, sensorial identity from this beer, but from microorganisms this all happen. So this probiotics not just confer functional properties from this beverage or health benefits, but also can confer different sensorial experience. So that's it's interesting to produce even more different beers. So, in conclusion, our data corroborate with the fact that probiotics, especially the strains Lacticase Bacillus paracase F90 and Lacticase Bacillus paracase 431. So, our data show that these are good candidates for to be used in breweries, as they may have several defense mechanisms such as membrane adhesion and H plus pump. Our analysis from proteome show that a lot of these um, proteins are really expressed. For example, this H plus pump, because it is important from surviving of this microorganism because they alpha acids from the whole, even in not so much concentration, can perform bacteriostatics effects in this bacteria. We make that this acid go inside to the cytoplasm from the cells and block every kind of protein synthesis or growth mechanism, which make this how the name suggested that the bacteria stays static, not performing any kind of metabolism, even the reproduction of of the cells. So our formulations show a probiotic viability between 5 to 8 log, equivalents of CFU for ml, 8 and 11 log equivalent of CFU per 7 portion of 350 ml through the manufacturing stage. So 350 is it's almost the countries is just about of a beer or a so it's from our kind of word. I know that in some countries more and but not less. Then this is a good portion per day. In addition, the expression of horsey gene seemed to be involved with microbiome resistant in control and passion fruit juice added formulations, while Heat A was only important in formulations with the addition of peach juice. However, as several other studies suggest, these genes may not be determined from microbial survival in beer. So our data show that it's not this specific gene or another specific gene that confer surviveness in the beer, but that's kind of can be in sometimes that gene and sometimes another, but it's important to be some kind of a pump or adhesion protein that which confer advantage to survive for this bacteria. Proteome analysis indicate that strain L431 may be the one of the best adapted to beer condition. Flavor analysis indicates that the strain used may interfere with flavor and odor. So the best was L431, but almost for all these strains used here, showed good results for our in our data. So it's good to use this. You can use this any way from brew a Catharina sour on a kind of sour beer. So I would like to thank my team in University of São Paulo in Brazil. I would like to thank my team in Technical University of Applied Science in Germany. So, so also Christian Hansen to provide the strains that we use it for our study, and Escola Superior de Cerveja e Malte in Blumenau, Brazilian, that our to share their knowledge and to give some technical support. And I will I'm let my email here, Marcus dot Herkenhoff at gmail.com. So you can, if you have more questions or more doubts, or also jobs opportunity to share or to make some kind of exchange with our institution. So let me know and send me an email. Thank you for everybody for this presentation to be selected for this award. I'm really proud of that, and I'm also really glad. And I hope that uh, this kind of lecture was interesting, and so you have more questions from this really wonder world that is beer and the omics application in this field. So the omics can be applied in so a lot of expertises and fields. So thank you so much, and see you soon.